freeze.
Right.
Yeah.
So, okay.
Even then, the oyster mushrooms that you'll find on dead logs and like the tree ear mushrooms, the Orobasidiomycetes, those are okay with freezes actually. And so you can still find those all winter long.
Okay.
Out and about. But yeah, the Asian market is a backup. I knew this might be an issue, so I'm going to leave that up to you.
Okay. Thanks.
And they're tasty.
Yes, they are.
I just got to put it out there. After going to the Asian market, I did eat them, but I do want to note that was before I put the ink on them. Just in case anyone else runs into that same problem.
That's good.
It's pretty obvious, but just throwing that out there.
All right. Any other questions about lab stuff?
Yes, I've got a question, Dr. Zong, about I think I heard you wrong one time. But so you know the lab one we have to do where you have to make them meet in the middle of the petri dish or whatever?
That was optional.
Okay, because I thought it was optional, but then I was talking to one of the classmates and we were like kind of not really certain of how that was.
Yeah, I mean, I would like you to. I'm not going to be grading on that because it's not guaranteed to work at all.
Okay.
It's really you're rolling the dice on that. That the two separate cultures that you pick have compatible mating types. Mm-hmm. So it's just if you do want to find zygospores, that's the way to do it.
Okay.
Yeah. Anybody else?
Can you hear me?
Oh, yeah.
I made my way over to mushroom TikTok.
There is such a thing?
There is such a thing. I tried sending you a video, but we're not friends on TikTok, so I can...
I'm too old for TikTok, I think. I think that's the rule.
But I found your TikTok.
Yeah. I signed in to watch some skateboarding videos once, and then that was it. Anyhow, so there's mushroom TikTok.
There is mushroom TikTok. I saw a video where someone, the one I was going to send you, it was about the puffs. She found one that was the size of her head.
Oh, yeah. They get humongous, and you could feed a family on them. The puffballs, kind of the rule for whether they're edible or not is if the spores are purple on the inside, then don't eat it. If it's pure white all the way through, then it tends to be pretty tasty and good. So you can go out to cow pastures and stuff and find them basketball size, watermelon size. They're pretty nice. That might be another option looking for fungi is maybe if you're not having luck in the woods, go to a horse pasture, right? Looking at cow pie, you will find fungi growing in there. Not necessarily illegal fungi. Those are actually quite rare in arid regions like this with cold winters. So you're probably safe picking those without getting into any legal trouble. So cow pies, horse pies, all kinds of animal dung. You'll find stuff. That's why we signed up for mycology class, right? To crawl around digging through cow pies for an assignment. But it is effective. Well, last week we got an intro to medical mycology. Today we're going to kind of transition into plant pathogen mycology. So let me bring up my screen. right. So, plant pathogen mycology is a way more developed field than medical mycology. It gets tons of grant funding. There are thousands and thousands of people with PhDs in plant pathology who are mycologists. We'll see why that is. We'll take a look at some of the impacts of plant pathogens and then just kind of get to know the disease triangle and meet a few of the most wanted plant pathogens. So we're not going to go too in-depth into this. We've got an entire course called Plant Pathology here at UVU if you're interested in plant pathogens. Sometimes taught by me, probably taught by Dr. Laney in the future, since he is an actual plant pathologist. Okay. Anyone know what this is a picture of?
It's a picture of humans.
Yeah, but it's a famous picture of humans in American history.
Would this be?
Not the Scarlet Letter, is it?
No, same time period though. You're on
Salem witch trials.
Salem witch trials. That's
There we
right. Salem witch trials has nothing to do with plant pathology. I just like this picture. Just kidding. It probably has quite a bit to do with plant pathology. So here's a map of Salem. If you're not familiar with the Salem witch trials, it was 1692. They had a pretty bad wet winter, and then 8 girls got sick, had convulsions, incomprehensible speech, went into trance-like states, their skin started burning, and so the obvious explanation was...
Obviously, witches.
Obviously, yes. It was obviously witches or aliens. They didn't know about those, so it has to be witches, right? And so 20 people wound up being put to death in this mass hysteria. So things were a bit crazy. People were getting visions that were actually allowed in trial, right? So if you had a dream that somebody was a witch or you had a convulsion, then that could actually be used as evidence. Creepy hotel. This is October, right? It's Halloween time. So this is actually one of the houses still there in Salem, and you can go stay there if you're really brave, I guess. Right?
Psycho.
Yeah, you have to be a super freak show to want to go spend the night there. The reason I bring up Salem witch trials is because it has been hypothesized that it maybe wasn't witches, which might shock you, I know, but if it's not witches, then what was going on? So in 1976, Linda Caporal, yep, something like that, Caporal, plant pathologists and anthropologists basically looking at why people would suddenly start convulsing, seeing visions, and having burning skin, and hypothesized that ergotism was the cause. So really eating some rye that was contaminated by ergot, okay? It does cause hallucinations. It makes your skin feel like it's on fire. You'll have convulsions, that kind of thing. It pops up after long, wet winters. That's favorable for its growth on rye. Rye was the main staple crop of that region. They just had a bad, wet winter. And in at least 2 of the girls' homes, they had big rye stores, like kind of grain rooms. So doing a bit of history there, it kind of looks like maybe that's what was going on. Of course, we can't really be sure. We don't know that ergotism was the case. We would probably like it to be, because that helps us understand why people were being so stupid. But As much as we'd like there to be a rational explanation for stupid behavior, I present exhibits A and B. Okay. Sometimes people are just dumb, so maybe that's the case. But this is an intriguing hypothesis, essentially linking this little organism, Seps purpurea, to an otherwise semi-unexplainable famous event in history. You see some ryegrass. This big dangling business there is the fungus. Okay, so it's infecting this staple crop. It grows there. All right, next slide. I'm going to show you the life cycle. Somebody jump in and help. Yeah.
Is that considered a pseudo-flower?
No, a pseudo fruit, perhaps.
Pseudo-fruit.
This happens after the flowering phase. Yeah. And remember that in grasses, they do make flowers, but they're not insect pollinated. They're wind pollinated, and so there's no need to like attract insects. Right. But what you're seeing there is a Sclerotium. You remember that term from way back in the day. Okay, what phylum is Claviceps from?
So it says ascospores, so ascomyces.
Ding, ding, ding. Yep. All right. So the big chunky business here that looks like a deformed grain of rye, okay, that's actually the Sclerotium, and it'll overwinter. In the spring, it germinates or kind of comes back to life, essentially, and makes the ascocarps, the fruiting bodies here. So there's the stalk, and then they've got little parathecia surrounding the outside. Okay, so this is a pretty complex structure. It's simpler than some of the fruiting bodies we've seen. Some of the pyrannomycetes, you know, where it's just a parathecium sitting on a piece of animal dung, right? This has a bit more of a complex life cycle. But inside there, we got parathecia. Ascospores come out. They germinate. They land on the new growing spring foliage, okay? They can reinfect an entire field and then overwinter in the grain crop. So when you go and eat it, what you're getting is this. All right, so here's a bunch of rye. Sclerotium. If you buy rye berries, actually you do need to watch out for this, okay? Most semesters when I teach this class, we do mushroom growing as a group in the lab, and we do that on rye grain. And so I have always big orders of rye grain coming in, and they always have some claviceps in there. You don't want to eat that, okay? It more or less leaves the rye plant intact, but it's still pathogenic on the rye and definitely a problem for humans. So if you do eat it, yeah, maybe you can get like high for a little bit, but also it makes your legs fall off, okay? The historical accounts of what came to be known as St. Anthony's fire are really horrifying. And so this happened all through the Middle Ages. Nobody knew what was causing it, right? They ate rye bread, and that's what was actually killing them all. So St. Anthony was the patron saint of the people suffering from this. It became known as St. Anthony's fire. Okay. Those with the more medical bent might also be interested in knowing that it's tied very closely to the bubonic plague, right? So bubonic plague is a bacterium, that's Yersinia pestis. But communities that subsisted on more rye than wheat or oats actually had worse severity of the bubonic plague. Any thoughts as to why? Guesses?
I don't know. Has it like mutated to a more virulent strain because there's more production? I don't know.
No, so it's just people who happen to be eating more rye happen to have more ergotism, even in low doses. And having ergotism made the plague worse. They didn't really play on each other directly. There wasn't like a organismal interaction between the bacterium and the fungus. It was just the toxins that people were eating. It's because it decreases circulation, especially in your extremities. Okay, it affects livestock as well. Okay, so these are the symptoms. So you can imagine if you've got necrotic fingers already, the plague already has an opening into your skin, your immune system's compromised, all that good stuff. Okay, so this is still something that occurs today, ergotism. Um, Eastern Europe, parts of rural Russia, they still have outbreaks of ergotism that affect people, damage their fingers. They have to get hands and feet amputated, that kind of stuff. All from a little fungus parasitizing a plant. Outside of this kind of charismatic case of a plant pathogen, right? We've got plant pathogens becoming an increasing problem globally for things like food production. All right, so in this figure, this is New Zealand imports of plant products, okay, on the left, and then associated pathogens on the right. All right, so panel A here goes with panel E. So this is crop, like arrival rate per host species, crop introductions or imports, and here's the arrival rate of pathogens over time. What do you see about these patterns? What's the relationship between the amount of crops they import, the amount of crop pathogens that are arriving?
More imports, more pathogens.
Well, sort of. Imports have actually dropped a bit, and pathogen rise is nearing exponential.
Oh, yeah.
And here in forestry products, like lumber, paper towels, that kind of stuff, their importing has increased dramatically, and yet pathogens have declined. With fruit trees, it's more or less stable, a slight increase since 2000, and yet fruit tree pathogens, steady incline. Pasture, okay, so grasses, that kind of stuff. In the 1960s and 70s, it was really high. They realized there was a problem, and so they stopped importing stuff, started relying more on native pasture products, plants, and yet pasture diseases are still showing up.
It just seems to me that when they get imported, those funguses take root in wherever they're being shipped to, and then they're not taken care of. People don't realize that they're here. So
yeah, that might be, it might be as simple as that, is like, oh, they showed up. It's not like they're going away now. Even if we stop importing crops that have pathogens on them, the pathogens are already here. And so it's kind of like once they're there to stay without massive effort to wipe them out.
it kind of reminds me of like the zebra sneller, I think is what it is, where it gets on the boat, they can
Zebra mussel.
and they just take over the ecosystem wherever they're at.
Yeah, they'll kill an entire lake.
Yeah.
Same thing happened in Utah Lake here with the imported carps, right? So a few people released their goldfish and some carps from their backyard koi pond into Utah Lake, and that was it. That's all it took, right? The native fish populations are essentially gone now. It's just carp and rotting algae.
Right.
So that's probably what's happened here is nobody was screening for pathogens back then. So they imported a bunch of stuff like, hey, let's get some apple trees, right? Those are coming from England, Armenia, United States. those apple trees came with pathogens already inside their tissues, pathogens that otherwise weren't there in New Zealand. And so now they've got fruit pathogens in New Zealand where there weren't any before. Okay.
So why for the forest diagram?
That was going to be my next question. Why indeed? They're
Yeah,
importing
so-
more forestry stuff.
Along with that, it looks like even so after they stopped importing, right, for the pasture, it's still increased. Is that saying that there's new species that they're finding, or what is that y-axis?
Yeah, new species they're finding per host species. So it's kind of like a proportion of how many pathogens per host. So they call it arrival here. So they're either showing up still, or we're detecting them newly for the first time as we go through time.
Is it possible that the imported ones are somehow interacting with the ones that were already there to make new species?
I think the time scale is probably way too short for speciation.
Okay.
Yeah, I mean, we're just talking a few decades, whereas it would be thousands and thousands of years for real speciation in fungi. But yeah, what is with the forest?
Or maybe like just as they keep bringing in new ones, there's enough competition that the new ones are winning out, and it's just staying level. Like that some species are being taken out of that environment, and because they're not competing well.
Maybe. One difference is that New Zealand has a pretty unique ecosystem, and in terms of the species of trees that make up their forests, it's different than pretty much anywhere else on Earth. I mean, they've got tree ferns, entire forests of tree ferns and Metrosideros and such. It looks like, something out of the Cretaceous period there still. They're not dominated by oak hickory or, that kind of thing. So very different host species are present in New Zealand, whereas for pasture, right, we're still growing the same pasture crops because that's what works for raising sheep or cows. Okay. Another thing is that, in the 1970s, they noticed this was a problem, and New Zealand has always been very big on conservation of their natural habitats. Less so worried about pathogens in, the fields or the fruit trees, but their native forests are found nowhere else on Earth. If all the apple trees in New Zealand die off, no big loss to biodiversity. But if the forests of New Zealand die off, that's a major loss of biodiversity. So they try really hard. They have very heavy inspection programs for imported forestry products, and that appears to be paying off because the pathogen load has dropped since they instituted those. Okay. Here's another one. This is the Big Island of Hawaii. And starting about 10 years ago, a new pathogen was noticed. It's a pathogen, and it happens to be really good at killing off these ohia trees. Quite good at it. It kills them pretty quick, and so it's called rapid ohia death. So the map in the bottom right, we're looking at confirmed outbreaks through time. It's worse now in 2020, but I haven't updated this map. So you can see it starts spreading here on the east side and just quickly grows out, basically taking over large swaths of the island. Okay. 85% of the Big Island is essentially covered by ohia trees. This is the native genre of plants in Hawaii. Well, of the woody trees at least. Kind of their dominant forest is ohia. It's specialized, early colonizer of lava fields, so makes sense. This is wiping out entire forests in a space of 10 years, just from this one fungus. That's a problem, all right? How did it get here or there?
The imports.
Yeah, probably. It probably came in with ornamental plant imports.
Yeah.
First on Oahu and then kind of spreading to the rest as they made their way out. So one little mistake and we're losing 80. That's
That you can do to try to protect the.
They're trying. I was involved in that to a small degree when I was there. Nothing's really working because now that it's on the island, it's actually spreading through insect vectors and wind. It causes the branches to kind of start twisting up, and that like twisting cracking motion ejects spores into the wind, and so it's looking pretty dire from the last I've spoken with people about it. Essentially the only hope is that at least some of these things will be genetically resistant to ceratocystis and will help recolonize the entire island, but we're probably going to be losing thousands of years before the whole thing's developed into mature forest again. Yay, good news, right? All right. So here's another reason why people study plant pathology. This is just a table looking at BT corn. Anyone know what that is?
Is that the recombinant product that's resistant to Roundup?
Not resistant to Roundup. That's actually Roundup Ready corn. You're on the right track. This one, they've added some bacterial genes to it. There's a bacterium called Bacillus thuringiensis that will basically kill insects. And so in organic farming, they're allowed to just take this bacterium, grow it up in a big culture, and then spray that bacteria all over the corn. With conventional farming, using transgenic corn, they actually just take that one gene from the bacterium and make the corn express it itself. And so insects who bite this corn, unless they're super resistant, tend to die. And so it's kind of like insect resistant corn. So there are a lot of examples of, so like Roundup Ready also, I just couldn't find the same nice table of crops, especially staple crops, wheat, corn, soybeans. These crops are increasingly being spliced with useful genes from other species to resist plant pathogens like insects, bacteria, fungi, that kind of thing. But this is showing just kind of the scale of the dollars involved here, right? And so we're looking at like the added dollar yield, okay, for each year just from BT corn. Sometimes that pays off, sometimes it doesn't, depending on pathogen load. But this is a multi-billion-dollar a year industry of plant pathogenicity. Okay, and
do those pathogens have any effect on humans now, or just on insects?
Just on insects. Yeah.
Just on insects.
Yeah.
At
heavily
least
tested.
that we've seen so far, right?
Well, went through FDA approval for 15 years, and you've been eating it for essentially your whole life, depending on how old you are. If you were born in the 90s or later, you've been eating this stuff your entire life. So the odds are pretty slim that there's any effect on humans.
So, how do we put that into us to make mosquitoes die when they bite us?
We could. We actually could do that. There happens to be a bit of an issue, though. Messing with human genomics is generally frowned upon by most people.
But there's no laws against it yet.
There are.
There
Yeah.
are?
Yes. Even in China, there are laws against it, but that's where it happened first. I don't know if you remember.
Oh yeah, I read the papers and the articles and everything.
Just a couple years ago, the mad scientist who made human babies immune to HIV.
Yes.
Which is awesome. I love it.
But it opens the door to something I don't think we're ready for yet.
Mm-mm. We're not good enough at this to be messing with human lives like that, especially because those kids are now going to pass that on through their germline. It's part of their DNA. So he didn't just alter them; he altered all of their offspring also. So
and that's the big thing, right? Like, can't we we're working towards messing with people, but not with their germline? Like with using viruses to inject different DNA into
somatic cells.
Yeah.
Yeah. You can't be sure that it's going to stick to somatic cells, though, can you? Viruses tend to be unpredictable. I think it's probably a bad idea at this point in our understanding of how genes actually interplay with each other to create a phenotype. I'm not sure that we're good enough to say with enough certainty this gene will give this phenotype, and that's all. Now, in corn, who cares, right? We're not like worried about corn suffering. If it doesn't work out, we just burn that field, call it good, try again. Doing that with people is something we don't want to go down that road again. We went there in the 1930s in Nazi Germany, and as you might know, that was evil. And so us trying anything like that again will probably be looked on as evil by scientists of the future because we don't know what's going on. That's my take on it. Where were we? I'll go off on a tangent about human gene editing.
We had just started. Yeah.
Okay. Yeah. So this can apply to human disease as well, but really the disease triangle concept was invented for plant pathogens. The idea here is that there can't be disease without all three of these items. A host that is susceptible, an environment that is favorable to disease, and a pathogen being present there. Okay. So if you're a plant pathologist, most likely your goal is to reduce plant disease or eliminate plant disease. This is why they make big bucks at like agricultural firms. It's economically important. We're trying to feed the world also. They look at this disease triangle as like a roadmap in a sense. Okay. So if you can reduce or eliminate, knock out one corner of this, it's going to reduce the pathogen burden on a given crop, okay, or a given forest. Okay. So you can knock out the presence of the pathogen. That, to me, that's like the most straightforward. No pathogen, no sick plant. Okay. But sometimes that's the hardest one to actually accomplish. You can change the susceptibility of the host. If the host isn't susceptible, no disease. Okay. If it's less susceptible, less disease. Okay. Or you could change the environmental conditions that favor pathogenicity. So if we look at our claviceps, our little ergot, okay, you could remove all the spores from the fields where people are growing rye, and that would get rid of ergotism. You could give them rye that's been genetically altered to resist being infected by claviceps. Okay. So that would get rid of ergotism also. Or you could have a less harsh, drier winter, and that would also reduce ergotism. So which do you go for as a plant pathologist?
Could you also speed up how fast it grows so the ergotism isn't mature before you harvest it?
Maybe. I mean, the alkaloids are still present. It's not exactly that little like sclerotium that's the problem, because remember the mycelium is growing all through that stuff already, and it's making that alkaloid consisting of things like lysergic acid, which is similar to LSD, that kind of stuff. So it's already in, even if you don't see it, it's in the grain. So maybe you might be able to reduce human disease that way, but definitely not the extent of plant disease necessarily. But maybe where you were going, and I just thought of this, is if you do prevent it from developing its sclerotium, like you're harvesting before that point, then that would remove the spore source for the next spring, right? So in that way, you'd be basically chopping away at that side of the disease triangle, saying, okay, we're removing the pathogen. So if you remember back to our like rust discussions when talking about basidiomycetes and their crazy dual host life cycle, getting rid of cedar trees is the best way to prevent your apple trees from being rust.
Yes.
You're getting rid of the pathogen. So sometimes that is the nice straightforward way. As we'll see, especially next week, not always the case, because as you might have known, fungal spores are pretty tiny and they get blown around by the wind. And so it can be a tougher challenge than you think to get rid of those things. In a greenhouse environment, sure, you can change the temperature, the humidity, that kind of thing. And so horticulturalists and people who like grow hothouse tomatoes and provide a lot of our food that way, they're well familiar with that, and they know what conditions are favorable to certain diseases, and so they can just bump up the humidity or bring it down if they notice a disease starting to show up in their greenhouse, and that can sometimes just get rid of it. No need to go spraying or anything. And then the other thing is this is, I think, where plant pathology has been going kind of recently, is to just focus on host resistance, right? Because you don't have tons of control over the environment, especially in crop fields, right, or the forest. It's going to do what it's going to do. Controlling the presence of the pathogen can be kind of hard. You can go in and spray, right? That gets rid of the pathogen sometimes, but pathogens are getting like resistant to those antifungals, just like antibiotics in medicine. And so if you just make the host resistant, it kind of, you don't have to worry about the other two. Does that make sense?
Yes, it seems like that's the one that we have the most control over.
It does feel that way sometimes, especially in like a natural setting. So in, you know, a forest, or especially in like a field of oats, right? The most control you're going to have, you can't stop the wind from blowing in spores. You can spray and spray and spray fungicides, and that might help you, but it's not going to for long, because they'll just develop resistance. So maybe we just go ahead and shell out that extra 2 cents a pound and get the Roundup Ready seeds, you know, or the BT corn, that kind of thing. And so I think that's the calculus that's been going on in farmers' heads recently, is like, this is something I can control. It does cost a bit for these seeds, but if I don't have to spray all the time, if I don't have to worry about like a strangely wet summer destroying my whole crop, then it might be worth it.
Yeah, wouldn't it always be worth the investment? Because if you don't and you lose, like, you know, a good portion of your crop, that's going to be more than the 2 cents a seed.
Yeah, for sure. You know, it depends on what your customers want, though. And so if you have customers who are insistent that they don't want any transgenic foods in their food supply, then you can charge more for that. Knowing that you're going to lose half your crop, you can basically double the price of it and make the same amount of money. So that is not to say that that's the only way that organic agriculture happens. They do spray a lot of stuff on their fields, copper sulfide and stuff, that are really horrible chemicals that kill off all fish downstream and stuff like that. But they're quote unquote organic, so they're fine for you. Don't worry about it. But those who truly follow like biodynamic practices, aren't spraying their fields, they plant twice as much and charge twice as much, and that's how, because half their crop is going to be lost to pathogens of some sort or another. It's just a problem with monoculture. So here's one outbreak in 1 year in the Columbia Basin. So southeastern Washington, kind of Oregon area. Okay, this is Phytophthora, I believe was, yep. So potato late blight, Phytophthora infestans. And this is 1995. There's a sudden outbreak, and that entire region, the state of Washington, lost $30 million out of their economy just from that one little outbreak, killing off potatoes. Okay. Idaho lives in perpetual crippling fear about Phytophthora infestans. That's the disease that caused the Irish potato famine. It's why we celebrate St. Patrick's Day in the U.S., right? Because people had to flee from Ireland, and the U.S. was willing to take them. Even though we were terrible to Irish people for a long time, we did take them in. So Phytophthora infestans, $30 million, one outbreak in 1 year. Here were the other outbreaks. Okay, so this is 2017. You can go online and just search for late blight map, and you can get basically like the weekly report of where late blight is being reported all across the United States and Canada. So this is hundreds and hundreds of millions of dollars of damage happening from this one pathogen, late potato blight. So I expect people are pretty interested in it. There is incentive for our government to be interested in it, to provide funding to try to stop it, because it's economically damaging to the entire state. So a lot of research interest, and we've got some interesting software that's come out not too long ago. It still kind of looks like it's running on Windows 95, but it's modern. This is called Late Blight. All right, so the Late Blight software, it taps into local weather reports. Okay. It taps into local outbreaks from the late blight map, so it sees where things are, calculates the prevailing wind direction. Farmers can log in and report their pesticide applications. So, hey, we're spraying for late blight here in our field, and you kind of register it. And so it takes all that into account, the variety of potatoes you're growing, how many per acre, all that good stuff. And it basically tries to predict risk in your field for late blight. And tells you whether you need to spend money spraying or not. What do all these things make you think of? The rainfall, prevailing wind direction, temperature, whether late blight has been
kind of goes back to like if it's favorable environment, susceptible host and pathogens and stuff like it. Just all of it combined.
They're looking to see if the pathogen is nearby, if the winds are pushing it your direction, if the environment is favorable, whether or not your crops are going to be susceptible to that particular strain of it. It is the disease triangle, just in a piece of software. You can download this and play with it. It's free. Runs on Windows. They made it free using a government grant, just because it's so useful to farmers. So if you're growing potatoes, you can have this on a laptop and basically map out your week and say, we'd better spray, right? And you do the math and you think, okay, well, that's going to cost us $850 to spray our fields, but the risk is pretty high that late blight's coming in, which means we lose everything. Okay, so helps you evaluate risk as a farmer. There's a bunch of plant pathogens, thousands of them, but there are certain ones that are like more infamous than others. Okay, so there's an interesting paper in Molecular Plant Pathology, which basically went out and surveyed all the authors who have ever contributed to molecular plant pathology and asked them to vote on what they thought like the number one most wanted pathogens were. And so here's our list of top 10. Okay, we're just going to take a brief look at the first three: Magnaporthy, Botrytis, and Puccinia. You've actually seen Puccinia before. But here's the Orzae. Yep, rice. Okay, this by double digits was the most important to all the readers because of how many people depend on rice as their staple crop. Okay, this is keeping billions of people alive, and this disease is all over the place. Okay. So, what kind of fungus do you think this is? What phylum?
Is it another Ascomycete? Or no?
It is. Yep. Pictured in the bottom right here is something kind of cool that it does, though. It will grow along the surface of the rice leaf, the blade of grass there, and find a stoma, right, where they open up to let CO2 in and release O2. It can detect that physically, and it will grow down into it that way. So here it's like growing down into the stoma. It grows invasively, okay, has lesions, it sporulates, asexual, boom, takes over entire fields, and it can infect the leaves and the heads of the rice, wiping out tons of fields, leading to starvation and death. Okay. So it does receive a lot of attention, research attention, but the scientists who study plant pathogens overwhelmingly said this is the one that needs funding, more attention. We don't know how to control it well enough. Okay. Number 2, Botrytis cinerea, the gray mold. This one was deemed to be really bad, not because it's infecting like such a staple crop, but it's because it can infect over 200 important plant species. So it just like can show up. It's not a specialist, it's a generalist. It shows up and just like start killing plants. It's a necrotroph. So it gets in there, immediately starts telling the plant to kill itself, right? The programmed cell death, it co-opts those pathways, kicks them on, apoptosis kicks in, the plant starts killing itself, and then it feeds on the carcass, all right? We spend €1 billion a year as a global community trying to control this. to greater or lesser extent. Okay, it's just wiping out tons of our food. Upwards of 10% of the global food supply is just destroyed by this every year. Okay. And then third, Puccinia. These are basidiomycetes, and they grouped these three particular species together: Puccinia graminis, Striaformis, and Triticina. But they all do infect wheat. Okay, so you can see them here. They wipe out wheat fields. It's a major pest crop in the United States where we grow tons of wheat. Okay. They are biotrophs, and their ecial host is barberry. So what's that mean for disease control?
It's probably a lot easier to get rid of the barberry than it is to get rid of the wheat, considering we kind of need the wheat.
We need the wheat. We definitely need the wheat. And so in 1918, we started a major program to eradicate all barberry in the United States. Same thing happened in England. We did it together. It was like a joint agreement. And they're still doing it. We stopped in, I think, 2006, eradicating barberry. We just decided it was too expensive, or we forgot the problem this was or something, but it is coming back in a big way. We've seen resurgences of Puccinia and massive declines in wheat revenues over the past about 15 years. I wonder why, right? So this was, and still is in England, considered one of the greatest achievements in plant disease management ever, okay? Up there with like eradicating polio, okay? So rusts are actually typically easy to control because they need those two hosts. You just get rid of the other host. Right. Plant pathology is a pretty rich field. We've just barely scratched the surface of it. So I'm just showing you like a brief glimpse at the table of contents from like an intro to plant pathology book. All right. So we have already mechanistic hypotheses about how fungi get into plants, the exact small RNA molecules that they use to trigger plant host defense, which causes the plant to go into overdrive and basically freak out so they can attack it in other ways. We've got coevolution theories that are well supported by phylogenies and genomics. So it's a fairly mature branch of mycology, especially compared to like medical mycology, where we know a whole lot about 4 or 5 fungi. We have no concept of what's going on with the human microbiome whatsoever. We just found out that fungi live in our lungs asymptomatically, essentially our whole lives. Like, what are they doing in there? We have no idea. Whereas plant pathology is a much more mature research field. Okay, it does tend to be goal-oriented, so it's an applied field. Obviously, it has inherent value built into it, and that value is we want to stop diseases from killing plants. Okay, so I am not a plant pathologist by any stretch. I am a fungal ecologist, and I had a crazy idea to manipulate the microbiome of plants to try to stop disease. I got the idea from looking at some medical studies where they did this with humans and bacteria, right? The poop transplants. That freaked me out when I heard that you could like eat somebody else's poop essentially, and it would cure a disease in you. So I decided to try it with plants. We actually got it to work. It's still a brand new field though, the idea of manipulating this. And so we're at kind of a point in plant pathology right now where the disease triangle is turning into a disease square, and we need to take into account the microbiome. So I think that's where we're headed right now. There are some reviews, I'll link to them online, that kind of point in that direction. plant pathology has spent a lot of time at the genomic level, at the mechanistic level. How are these things getting into cells? How are they interacting with the host genome? That kind of stuff. Less attention has been focused on how are they getting around in the first place, and what are they encountering when they land on the leaf, right? So if there's another species there altering it, the microbiome that was already present, that may be something we need to take into consideration. So that's as far as I want to go into plant pathology lecture-wise with you. We're reading a pretty interesting paper about flower mimicry and some rust, and we'll have our quiz about that before moving on in this course. So we got a bit of time for any. Anybody? Nothing?
I have a question just about the quizzes in general.
Okay.
I think on your syllabus, I could be wrong. It's been a while since I looked at it, but it says we only have like 10 quizzes or something. Is that accurate? Are we just going to stop having quizzes at some point?
That's accurate. Any over that, if you remember, are going to be bonus.
Oh, sweet. Okay. Thank you.
Yeah. The same sort of scenario for lab assignments, right? We're going to actually have more than the minimum number of points of lab assignments, but doing those will be bonus points.
Cool. Thank you.
Mm-hmm. All right, well, we can end a few minutes early. I'm cool with that. I will get the quiz posted in the next hour or two, and see you guys next time.
Next week's fall break, right?
Is it?
I believe it is. I think it's Thursday and Friday of next week.
So we only have Tuesday?
Yeah.
Okay. Wow. Every day is the same. You know, I just live in a bathrobe now. Time is a flat circle. I have no idea what's happening. Okay. I'll check in on that.
I didn't know it was until this morning because my kids were saying something about it.
Okay. I may have to like rearrange a quiz for us, that kind of thing. But thank you for letting me know.
Yeah, no problem. Have a good one. Thanks.
Yeah. See ya.
Thank you, Dr. Zahn.
Adios.